Hey everyone, and welcome to this Masterclass Explainer. I am so incredibly excited to be your virtual O Level Biology tutor today. Seriously, if you're gearing up for your exams and maybe feeling just a little bit overwhelmed by all the crazy chemistry happening inside living things, take a deep breath because you are in exactly the right place. Today, we're going to do a rock-solid, comprehensive review of everything you need to know about biological molecules and enzymes. We are diving straight into the source material, breaking down every major concept, every single minor detail, and all those tricky diagrams so you can walk into that exam room with absolute total confidence. Let's get into it. Here is our game plan for today's session. First up, the molecules of life. Second, chemical tests. Third, enzymes as biological catalysts. And finally, temperature and pH effects. Okay, let's dive into this. Section 1. The molecules of life, building blocks from the ground up. So, starting with the atomic foundation of biological molecules. You can't build a house without knowing what your bricks are made of, right? Notice how this breaks down exactly which chemical elements make up our key nutrients. This right here, it's an absolute goldmine for your multiple choice questions. And as your tutor, I want to give you a direct study hack right off the bat. Actually, scratch rote memorization. Let's just look at the pattern together. For carbohydrates and lipids, just memorize CHO, carbon, hydrogen, and oxygen. Just note that lipids have a much lower oxygen content compared to carbs. Now to get to proteins, you just take your CHO and add nitrogen, or N. Sometimes proteins have sulfur or phosphorus too, but N is the big one. And for DNA, it's CHON plus phosphorus or sulfur. So just remember that sequence. Start with CHO, add N, then add P. Now let's think about how these elements actually come together in the real world. Think of it like snapping together tiny building blocks. Those smaller, repeating subunits are called monomers, and they link up in a process called polymerization to form giant polymers. For instance, amino acids link together to form incredibly complex folded proteins. Fatty acids and glycerol combine to make lipids. Sugar residues, or monosaccharides like glucose, link up to form large carbohydrates. And nucleobases form nucleotides, which literally stack together to build that incredible double helix of DNA we all know. It's like a giant puzzle assembling itself. Let's look a little closer at these structures, because Cambridge absolutely loves to test this. First, amino acids. There are about 20 different types in animal proteins, and they link together via what we call peptide bonds to form very specific sequences. Then, think about complex carbohydrates. Imagine glucose monomers forming 3 different massive molecules. You've got starch, which is how plants store energy, glycogen, which is how we animals store energy, and cellulose. Cellulose chains actually form these straight microscopic fibers that layer together to build super tough plant cell walls. Oh, and here is a crucial exam detail to lock in right now. Cellulose, starch, and glycogen are all completely insoluble in water. All right, moving on to section 2, chemical tests and detection, proving what's inside. So we know the building blocks, but if I handed you an unknown food sample in a test tube, how would you prove what's inside? It is time to shift into investigator mode. You absolutely must know these chemical tests and their precise color changes for your practical paper. Let's rapid fire through the first 2. Testing for starch? Super easy. Shake a small amount of powder in warm water, add 3 or 4 drops of iodine solution. If starch is there, boom, it turns a deep blue or blue-black. For protein, we use the Biuret test. Pay attention to the exact volumes here. Take 2 cubic centimeters of a protein solution, like 1% albumin, add 2 cubic centimeters of dilute sodium hydroxide, followed by 2 cubic centimeters of 1% copper sulfate solution. If protein is hiding in there, the whole thing turns this beautiful purple. Do not mix up your colors on the exam. Now for the test that trips up the most students, Benedict's test for reducing sugars like glucose and maltose. Picture a color gradient. You take 2 cubic centimeters of your sample solution, add an equal volume of Benedict's solution, and, this is absolutely crucial, you must heat it in a boiling water bath. If there's no sugar, it stubbornly stays clear blue. But if glucose is present, it progresses through green, then yellow, and finally it forms a totally unmistakable brick red precipitate. Why? Because glucose is a reducing sugar, and it literally reduces the solution to a red precipitate of copper oxide. And this brilliantly illustrates our final chemical test, the ethanol emulsion test for lipids. Remember, lipids don't dissolve in water, but they do dissolve in ethanol. So, step 1, shake a couple drops of cooking oil with about 5 cubic centimeters of ethanol in a completely dry test tube until it completely dissolves. Step 2, pour this clear solution into a second test tube that already has water in it. Instantly, the lipid drops out of the solution and forms this incredibly clear, milky white emulsion right at the top. It looks really cool in the lab. Okay, before we move on, I want to cement these tests in your memory. Seriously, say this aloud with me right now. Benedict's builds to brick red. Butyrate is purple for protein. And remember, iodine is blue for starch, and ethanol is milky for lipids. Lock those phrases in, and you literally just guaranteed yourself some easy exam marks. Awesome. Let's hit section 3, enzymes, the biological catalysts speeding up life's reactions. Let's shift gears a bit. We've got all these amazing molecules inside us, but how do they actually interact? I mean, how does digestion happen fast enough to actually keep us alive? Enter the true unsung heroes of biology. Enzymes. If an exam asks you to define an enzyme, this specific definition is exactly what they are looking for. An enzyme is a protein that functions as a biological catalyst. A catalyst increases the rate of a chemical reaction by providing an alternative pathway with a lower activation energy. But here is the absolute most critical part you must write down. It does this without being changed or used up at the end of the reaction. Because they aren't consumed, your body can use the exact same enzyme over and over and over again. To put that into perspective, a single molecule of the enzyme catalase can process millions of molecules of toxic hydrogen peroxide every single second. That is just unbelievable biological efficiency. Let's do a deep dive into how this actually looks. Imagine a tiny, highly specific Pac-Man. That's our enzyme. It has a specific 3D dent on the top called the active site. The molecule we want to react, the substrate, enters that site. When they bind together, we form a temporary enzyme-substrate complex. Being in this incredibly close proximity basically forces the chemical reaction to happen. Finally, the reaction finishes, the enzyme spits out the newly formed products, and the enzyme itself is totally unchanged and immediately ready to grab another substrate. Now, what's really interesting about this slide is how insanely specific enzymes are. They only work on one specific substrate. Why? Well, it's perfectly explained by the lock and key hypothesis. In this model, think of the enzyme's active site as a rigid lock. It has a highly specific three-dimensional shape. The substrate acts as the key. If the key doesn't have the exact right complementary shape, it simply will not fit into the lock. No fit, no reaction. This brilliant mechanism ensures your body doesn't accidentally trigger the wrong chemical reactions at the wrong time. Okay, listen up, because to get full marks on an enzyme action question, these are the 4 exact vocabulary words you must weave into your answer. You start with the substrate. It binds to the 3D active site of the enzyme. This temporary pairing forms an enzyme-substrate complex, and what's released at the end is the product. Use these exact terms in that order, and the examiner will literally have no choice but to give you maximum credit. Right, on to our final stretch. Section 4, environmental effects. Temperature, pH, and denaturation. This is frankly the most heavily tested area we're covering today. We know how enzymes work perfectly, but what happens when their environment changes? How do extreme heat or harsh acids break these delicate biological locks? Well, before we look at the data, you need to know how we actually investigate this in a lab. There are 2 main ways. We can measure how fast a product is formed, like using catalase to break down hydrogen peroxide and measuring the volume of oxygen gas produced over time. Or, alternatively, we can measure how fast a substrate disappears, like mixing the digestive enzyme amylase with starch and using our trusty iodine test every 10 seconds to see exactly how long it takes for the starch to be completely broken down. Now, let's analyze the classic temperature graph together. Picture the rate of reaction climbing. As temperature increases from 0 to about 37 degrees Celsius, the rate of reaction shoots up. Why? Because the molecules are gaining kinetic energy. They're moving faster and faster, leading to a much higher frequency of effective collisions between the substrate and the active site. That peak, right at 37 degrees, which, no surprise, is human body temperature, is called the optimum temperature. But look what happens immediately after you pass that peak. The rate doesn't just slowly taper off, it completely drops off a cliff. That steep drop-off, that is caused by a massive problem called denaturation. When you get too far above the optimum temperature, the excessive kinetic energy literally shakes the protein apart. The active site loses its specific 3D shape permanently. The substrate key no longer fits the lock, and the reaction stops entirely. Think of it like cooking an egg. The clear, runny egg white turns solid and white because the proteins have denatured. You absolutely cannot uncook an egg, and you cannot reverse heat denaturation. Once it's broken for good. And let's see how this builds when we talk about pH. Every single enzyme has an optimum pH, a specific level of acidity or alkalinity where it works its absolute best. It usually looks like a perfect bell curve on a graph. Deviating from this optimum alters the enzyme's tertiary structure.
For instance, the protein-digesting enzyme in your stomach thrives in a highly acidic pH of 2, while pancreatic enzymes prefer a slightly alkaline pH of around 8. Extreme pH will denature the enzyme entirely, though sometimes, if the shift is minor, it can be reversible if the core structure stays intact.
So here is your ultimate cheat code. The crucial point is knowing exactly what to write when an exam question asks, why did the reaction stop at 60 degrees Celsius? Here is your study hack summary list. You write, the high temperature caused the molecules to gain excessive kinetic energy. This broke the bonds, changing the three-dimensional shape of the active site. The enzyme became denatured, meaning the substrate no longer fits. Therefore, the frequency of effective enzyme-substrate collisions dropped to 0. Boom! Full marks every single time.
Wow, okay, we have covered a massive amount of ground today. You now completely understand the fundamental molecules of life, from glucose all the way to DNA. You've memorized the specific chemical tests and their color changes, and you've absolutely mastered the elegant lock and key mechanics of enzymes alongside the environmental dangers of denaturation. You have the knowledge. You have the exact exam phrasing. The only question left is, how will you apply this perfect fit to confidently ace your upcoming biology test? I know you're going to do great. Keep reviewing, keep studying smart, and I'll see you in our next explainer.